Okay, hello everyone. Thanks for coming back on time. My name is David Lobel. I'm a professor in Earth System Science here at Stanford, part of the Stanford Data Science, and I'm going to moderate this exciting session that we have. We have 4 lightning talks. By lightning, what we mean partially is that there's not going to be Q&A after each talk. We're going to save that for the end when all the speakers will come up, and we'll have about 15 minutes for Q&A at the end. So without further ado, we're going to get started with our first speaker, Sandrine Dudoit, who is a professor and chair of the statistics department at UC Berkeley, as well as in the School of Public Health and Epidemiology. And she'll be talking about learning from data in single-cell transcriptomics. Sandrine.
Thanks very much for the introduction, David. And I'd first like to thank also Chiara and the organizers for inviting me to speak today. It's my first in-person conference. It feels a little odd, but it's really nice as well, so I'm very happy to be here. So yes, I will talk about learning from data in single-cell transcriptomics and present a little data science vignette in a biological context. So my group's work in this area was motivated by a long-time collaboration with the NI Lab at Berkeley, now at the NIH. And it's on the study of stem cell differentiation in the mouse olfactory epithelium. So one of the goals of this study in the NI Lab was to elucidate the molecular and cellular mechanisms that underlie stem cell-mediated development and regeneration in the olfactory epithelium. And this work has important implications in medicine, in particular to the prevention and treatment of neural tissue damage and degeneration. So think, for example, Alzheimer's disease. So our collaborators in the NI Lab focus on the differentiation of a particular type of adult tissue stem cell, the horizontal basal cell. And they use the p63 protein as a molecular switch. So under normal conditions, the HBCs will self-renew, but when p63 is downregulated, they will start differentiating into a range of different cell types. And we use single-cell transcriptome sequencing, or in short, RNA-seq, to measure the expression levels of, on the genome scale and at the resolution of single cells in mice that were normal, wild type, and also mice that had the p63 gene knocked out. So this is an illustration of the different types of cells in the olfactory epithelium. We have the horizontal basal cells here in green. So normally they will self-renew, but when the p63 gene is downregulated, they will differentiate into a range of different types of cells, globose-basal cells, and different types of neurons, as well as subsyntaccular cells. So here's, in a nutshell, what single-cell RNA-seq does. So you first isolate individual cells, then you extract their RNA, you chop up the RNA, and you sequence the little fragments of RNA. So you find all the different bases, A, C, G, U, and then you map the small fragments back to the genome, and the idea underlying RNA-seq is that the number of reads that fall in a particular gene region gives you a measure of the expression level of that gene. So our entry point here comes after much pre-processing. There's image processing, there's base calling, and sequence alignment. And after these steps, we can summarize the data from RNA-seq in a matrix whose rows correspond to genes and whose columns correspond to cells. And the entries of the matrix are the read counts. That gives you the expression measure of, say, gene 1 here in cell 1. So single-cell RNA-seq studies gives you a great illustration of the range of questions that you see in data science, where you have complex data, where there are multiple steps of analysis, and where it's really important to have sound and statistical principles and methods to derive reliable and interpretable biological insight. So the data are complex in a variety of ways. Dimensionality, of course, we typically have large P, and either small n or large N. There's various levels of processing depending on where you enter on that analysis pipeline. You have various types of data in addition to the familiar matrix, XN by TP. You have image data, you have character string data, you have graph data. We have data on data. For example, annotation data on the genes and on the cells. There are large technical biases, for example, due to batch effect from the sequencing run. Also sequence composition effect, like GC content effect. There's often low signal to noise and sparsity. In single-cell RNA-seq, a particular cell typically has up to 80% of its genes that have 0 counts. So our approach to this single-cell RNA-seq study follows a general data science workflow where we first try to understand the domain context and question. Then we frame and translate the domain question into a data-enabled question about statistics. We translate the question into statistics. So this is a step that's often overlooked, but it's essential, and actually it's far from trivial. And in this context, I like to think about this quote from Tukey: far better an approximate answer to the right question, which is often vague, than an exact answer to the wrong question, which can always be made precise. So in our case, the main questions that we're dealing with are: what are the groups of cells that we see in the olfactory epithelium? What genes are differentially expressed between these different cell types? What are the lineages and where they branch? And also what genes are involved in the differentiation process. So then, of course, there's the important aspect of designing the study to make sure that the question of interest can actually be answered with the data that we collect. It's so common to see obvious confounding problems in these studies. We also want to answer the question as accurately as possible given available resources, so dollars, biological sample, reagents, time. EDA is really important in this context to understand our data, its main features, as well as detect obvious problems with the data, so for quality control, quality assessment purposes. So it's really crucial to look at data to avoid garbage in, garbage out. So no matter how good your machine learning method, if you feed it garbage, you're going to get garbage. Data cleaning and pre-processing are also really important, and they often have a larger impact on the results than the choice of downstream machine learning method. So we spend an awful lot of time at that step. This is the typical entry point and comfort zone for statisticians and computer scientists. This is where we spend most of our time. Why? Well, because the rest is actually really hard and somewhat unsatisfactory for us that have math backgrounds. There's no cut and dry mathematical answers to these questions typically. And there's somewhat of an art actually in applied statistics and data science. And we really need to be immersed in the scientific context and have also sustained communication with our collaborators to make sense of everything that's going on here. And it's really important, I think, as statisticians to be involved in these other aspects of the data science workflow, because this is where the domain meets statistics, and we need to make sure that there's no disconnect between the work we do here and what's happening in the science. So in the context of the actual machine learning or statistical inference, we want to make sure that the method actually answers the question and that the output of the method are consistent with how the results will be used. In particular, it's good to think about the meaning of probabilities in all these methods. Where does the randomness come from? What are these sampling units? So there's a few caveats here. So it's very often to see hammers looking for nails, unrealistic modeling assumption, and I like this. I always say it in French because that's my background. But why make things simple when they can be complicated? So often the simplest approaches do quite well in these contexts. So this is a highly iterative and collaborative process, and of course something that's crucial throughout the process is software, good and reliable software. So here are a little bit more specifics about the workflow we've developed to address the question from our collaborators, so to study the differentiation of stem cells in the olfactory epithelium. This is more like an advertisement of the work that we've done, because I won't have much time to spend on any of these topics. Also, I've indicated here in gray font, gray characters, the name of the R software package that we've developed for each of these steps, and they're released through the Bioconductor project. So before we do anything, we look at data for quality assessment purposes. We also spend a lot of time on normalization. The goal of normalization is to adjust read counts to make sure that when we see a difference in expression measure, either between genes or samples, that this difference reflects the biological effects of interest and not some unwanted technical effects due to batch or to sequence composition, for example. So we've developed a range of normalization methods, ranging from very simple methods that just do global scaling within a cell to more sophisticated method like regression on unknown factors of unwanted variation, which we obtain through factor analysis. So this is unsupervised normalization. And we've also developed a framework for applying and assessing the performance of these different normalization methods to have a data-driven way of selecting an appropriate normalization method for a particular data set. So a very common task in these genomic studies is to cluster either the genes or the samples. Well, as you know, clustering is a very difficult problem, and it's a frustrating problem because no matter what, the data or the clustering algorithm that we use will get clustered, whether they're real or meaningful, whether they're replicable or how we validate them is another story. So you might ask yourself, why yet another clustering method? So actually what we're doing is not to propose a new clustering method, but a framework for applying and comparing a variety of different clustering algorithms, and then aggregating the multiple candidate clustering into a stable consensus clustering. So it's essentially an ensemble approach for leveraging the strengths of different clustering approaches and formalizing what people often do in practice, which is try a bunch of different algorithms and sort of try to compare the results and combine them. So this is a more formal way of doing this. We've also developed a method for merging clusters in order to navigate the trade-off between the resolutions of the cluster and also their replicability across data set. So the rest of the methods that I'll mention are more specific to the trajectory inference questions. So we've developed a method called Slingshot for inferring the cell lineages and their pseudotimes. So the first step relies on a cluster-based minimum spanning tree to infer the global lineage structure. So the number of lineages, whether you have 2, 3, 4, 5 lineages, and also where they branch. So we work on the clusters and then fit a minimal spanning tree to these clusters. And then the second step is at the level of the cells. And then we identify pseudotimes for the cells along each of the lineages using simultaneous principal curves. So the pseudotimes are essentially the locations of the cells on the lineages. They correspond to their to transcriptional time, so not necessarily chronological time. So this method allows us to identify any number of lineages, and also we can supervise the inference of the lineages by telling it which are the terminal states. So in our case, we know that we have neurons and sustentacular cells as terminal states, so we can feed that information to the way we build the minimum spanning tree. And then the next step in the process, of course, is to try to understand which genes influence the differentiation process. So we've developed a framework for differential expression that relies on the continuous nature of the differentiation process. So for this, we rely on negative binomial generalized additive model. We can, by looking at the coefficients of the GAM, and contrast based on these coefficients, we can identify different types of differential expression patterns. I'm sorry, 2 minutes, okay, between or within trajectories. So I'll show you very quickly a few of the results. So we applied Slingshot to the clusters that we get from RSEC, our ensemble method. And so this is a 3-dimensional principal component analysis plot of the cells colored by cluster. We fit a minimal spanning tree. We find 3 branches, 3 lineages, and then the simultaneous principal curves. And then we apply this NB-GAM approach to identify genes that are differentially expressed within lineage and also between lineage. This is very quickly just the GAM. So Ys are the gene expression measures, Gs are genes, I are cells, Ts are the pseudotime, and we use cubic splines to parameterize the mean of the NB-GAM. And we look at contrasts of the GAM coefficients to fit different types of differential expression patterns. So this is the types of, depending on what contrast we look at, we can look at differential expression within a lineage, like these blue lineages, which gene expression increases with pseudotime, or differences between the blue and the orange lineage. So just a combination of linear, just different types of contrast between the betas. And we've applied this to our olfactory epithelium data set. And these is an example of the top 200 differentially expressed genes within the neuronal lineage. You can see different clusters of genes that have different regions of activity early or later on in the lineage. And this is an example of applying a test for finding genes that are differentially expressed between lineages. And here we've identified 4 transcription factors that correspond to epithelial cell differentiation. So I'll just jump very quickly to the results. I won't have time to show all of the results, but basically by using this GAM-based approach, we can uncover the transcriptional programs that are active in each of the 3 lineages and find both known and unknown and previously unknown marker genes. So overall, we can also use the GAM and look at derivatives of the GAM to look at which genes increase now in expression. So it gives us the ability to address a broader range of biological questions. So it's really a flexible approach to uncover the transcriptional programs associated with stem cell differentiation. So overall, we found that Slingshot and this trait-seq GAM approach revealed that olfactory stem cells use divergent strategies to generate the major cell types in the epithelium. On the neuronal lineage, we have numerous step-like transition, but fewer gradual changes in the sustentacular and the microvillus lineage. So I'm out of time, and I'll stop here. Thanks very much for your attention.
Thanks, Sandrine. So our next speaker this morning is Yuri Leskovitz. He's, as you know, a professor of computer science here at Stanford. He's also been a very active leader in the Stanford data science. So we all have him to thank in part for this Stanford data science existing. He's going to talk today about mobility networks for modeling the spread of COVID-19.
Super. I'm really excited to be here, and I'm amazed what has grown from what we started with Emmanuel a few years ago. So great, super excited. So what I'm going to talk to you today about, if it's going to show up, is actually a project that we started exactly 2 years ago. And it was my COVID lockdown paternity leave type project. So my daughter was born actually yesterday, 2 years ago. So this is what we've been working on, and I want to show this to you because it's a nice story. It will, the pandemic is now kind of over, so it will be kind of a good way to remind us what we went through, and I'll show you some results that we were the first to establish about how the pandemic spreads, how to reopen the economy, and so on. So that's the idea. So March 2020, right, exactly 2 years ago, the outbreak hit, and there were these drastic and severe measures, right? We went into the lockdown, social distancing, masks, we were disinfecting every piece of material everywhere, and so on and so forth, right? And then there was a huge number of unknowns, a lot of misinformation, disinformation. Here are some news articles. And of course, there was also quite a lot of protests later on, kind of people asking, do we really need this? This is worse than the virus itself, and so on and so forth. So when the pandemic started, in my group we actually did a brainstorming session and said, okay, how could we help? Can we stop what we are doing and help? So here's one example of a project that we did during the pandemic that actually had quite a bit of impact. And I would say that this is probably, in terms of number of human lives touched, I think this rivals, me building a recommender system for Pinterest with around 500 million people touching every month. This, I think, touched also a similar order of magnitude of people. So coronavirus, right, how do viruses spread? They spread through human contact, right? So the way you can think of this is that there is some underlying contact network that modulates the spread of the virus. And then, right, this virus starts somewhere, and through contact between individuals, it moves from one person to another, and this way how it spreads through the population. But the important thing here is that if you want to model the spread of the virus, you need to know what the contact network is, who is in contact with whom. And the problem is that this human-human contact network is basically unknown. So what epidemiologists or standard epidemiology tries to do, it says, oh, let's just assume the graph is fully connected, right? Everyone can infect everyone. That's kind of the most basic assumption. And that's one fact that makes this modeling hard. Another fact that makes the modeling hard is that the world is super dynamic and super non-stationary. So it's kind of changing all the time. And especially if you think about March and April of 2020, each day was brand new day in the history of the Earth in terms of how humans behaved, right? So it's very hard to build a model and then let it run because the world is non-stationary. So what we asked was, or what our premise was the following, was we said, if we can estimate the dynamic human contact network, then virus modeling will kind of be easy and will be accurate. And if we are able to do this, then we'll be able to ask and answer some fundamental questions like, is social distancing really needed, right? And for example, how should we reopen the economy as we ease back to normality? So the question was, how could we estimate hour by hour kind of real-time contact network of a population of, let's say, 100 million plus. And what we decided to use to do this was an innovative data source, which is basically the geolocation pings of different cell phones. So the way you can think of it, that basically for different cell phones, we know their locations. Right? And we had this data for 10 of the largest US metropolitan areas with combined population of around 100 million, and we had this data for every hour of the day. The data actually came from a company called SafeGraph that basically partners with application developers and then users of those application developers to give back the anonymized pings. And we also, not only that we know the locations of these cell phones, but also we would know, for example, where these cell phones are. And then we can look in other databases and know what is the square footage of that area, how long were they in that space, and so on and so forth. So what this allowed us to do was to build a epidemiological model that has two types of locations. It has census block groups. These are geographical areas between 500 and 3,000 people live, and points of interest, which are grocery stores, pharmacies, schools, churches, anything you kind of go to. And based on this, we built a very simple kind of SEIR type model where the graph of this, how people move, changed hour by hour. So basically for every hour of the day, we would infer how many people from a given neighborhood go to a given specific drugstore, to a given specific grocery store, to a given specific hotel. And we have about 500,000 of these points of interest and about 60,000 CBGs. And we use data from SafeGraph, US Census, from epidemiological literature, as well as from the New York Times. And just to show you an example how the model works, the first panel is mobility. The middle panel is number of infected people, and on the right-hand side is kind of how many people are infected in the Chicago metropolitan area. And you can see how, in the downtown you get a lot of infections, and then as people move around, it kind of bubbles up everywhere. So this is, and what is important here is that the model predicts who will get infected, right? What home neighborhoods people are from, and where will they be infected, at what point of interest. And right, we spent a few months basically working on this infrastructure and pipeline and the model, and the students were very worried. Is this going to work? Are we going to be kind of scooped and overtaken? What's interesting is that we our model only has 3 static parameters, which is probability of infection at home, probability of infection in a point of interest, and the initial fraction of infected population. And you can see that with these 3 static parameters, you can model very different trajectories in different cities in United States. This was the first wave of epidemic, and the X's, the dots, those are the measurements. That's the ground truth data. So you see the variance in those measurements or uncertainty is huge. Okay? So that's what we did, and now I want to give you some results of what was interesting. So the first question is, did stay at home really matter? I wanted to know this. Actually, it turns out, right, given how much we decreased our mobility, if we were not to decrease it, 32% of people would get infected in a single month. So here are different trajectories. Yellow is if we were moving around as if it was, end of February, and then the black is the actual mobility. Okay, so that's the first interesting observation. Another interesting observation is that you can now ask in what different points of interest do these infections happen. And here's the ranked list according to the risk, and it makes sense, right? It's places where people congregate close together for a long time, like restaurants, fitness centers, and so on. So these are all things that at that point were unknown, and we were the first to quantitatively demonstrate them. Another interesting thing you can ask is, how should I reopen the economy? So we were able to simulate what happens if you reopen restaurants at 20% of the maximum occupancy. What happens if you reopen drugstores at this other level of maximum occupancy? And this means you can very nicely trade off between the amount of business these places are going to get or they are going to lose versus the number of infections and the stress on the health system. And for example, if you would cap at 20% maximum occupancy, you would be able still to get about 80% of all the visitors, but you would only you would curb 80% of infections and you would lose only about 40% of the business. So these trade-offs our model allows us to do. And then another interesting thing was that we can also look at social disparities. In particular, poor were hit much more by this virus. Here I'm showing relative risk of infection between rich and poor neighborhoods in different cities. And the question is, why is that? And the explanation or the hypothesis at that point in time was that poor have so many coexisting conditions that then they get hit by the virus much more. Actually, what we were able to show through this data is that poor were able to decrease their mobility less than the rich, people from poor CBGs decrease their mobility less than people from rich CBGs. So that's one part of explanation. But there's another interesting part is that poor went to places that were more crowded and they stayed there longer. So a single trip to a grocery store, the risk of a poor person getting infected there was twice as much as for the let's say rich person, in a sense that they went to places where there was more people per square foot and they stayed there twice as long. Okay, so these are some of the interesting things we were able to build, and basically build a tool that allowed the decision makers to make, the public health officials to make decisions. We published this, we started the work in March, we finished in August, and then we worked very hard with Nature, and I think it was published in October. It was really work with 3 amazing Stanford students, Serena, Emma, and Pangwei. It was amazing in terms of the reception of these papers. We gave over 400 interviews and appeared on various TV channels at weird hours of the day. But what was more interesting, as I said earlier, is these results were really used by governments all over the world. There's an example of a screenshot from a Polish, I think, TV. Basically, that's the graph I showed you from our paper, but translated to Polish. In Japan, I heard that basically they showed our paper on TV and said, this is what we have to do. US state governors were referring to what we were doing, and even US Supreme Court briefings are citing our papers. We also worked with New York Times on a couple of visualizations that appeared through subsequent months for people to try to understand these risks. And we also built a decision support tool and worked with various counties and states to implement this for them to help them make decisions, especially with reopening the economy. So this is what I wanted to say and share a story about the research I was doing during the lockdown. So thank you so much.
Great, thank you, Yuri. Our next speaker this morning is Fiorenza Micheli. Fio is a colleague in the Woods Institute of the Environment. She directs a center on ocean solutions, and she, among other things, collects data in addition to analyzing data, and the way she collects data is often to go scuba diving. So if you're wondering who the smartest person in the room is, you can consider that. So her talk today will be on harnessing the data revolution for ocean health and sustainability. Fio.
Thank you. Thank you so much. And good morning, everyone. Today I will present very briefly on the huge potential of data science to contribute to addressing some of the great challenges in ocean science and sustainability, and hopefully, conversely, by addressing those questions, also sparking advances and innovation in data science itself. And Drs. Cohen and Nakayama are here in the room as well today. So I want to start by asking the question, why ocean? Why the focus on ocean? So there's at least three major reasons. First of all, oceans are really important. They are a huge part of the life support system on this planet. They provide food, nutrition, regulate climate, support coastal and maritime economies. The second reason is that all of these key functions and services are threatened by warming, ocean acidification, over-extraction of resources, pollution. And then, so we're losing them, and the impacts result in loss of biodiversity, impacts on human health, livelihood, and lives. And then the third reason is that oceans are arguably the last frontiers for discovery. We know actually still very little about oceans. Dave was saying, I'm a scuba diver. I actually had the privilege of doing ocean exploration on deep sea submersible. I've done many dives in Alvin, which is the submersible that went down on the Titanic. And no, it's a life-changing experience, and every time we find new things. So it's super exciting, but it's also very costly and very slow. And so there's a lot to be learned in ocean. It's really the last frontier. And what has happened in the last 2 decades, it's a true explosion of data from buoys, from animal-borne tags attached to tunas and sharks and seabirds, from autonomous vehicles. And in fact, it's estimated that more data have been collected in the last 2 decades than in the previous 100 years. So this is the beginning of the examples that I want to show, because we are really seeing a huge potential that has no precedent. And what I showed in that chart is just a fraction of what's available, because new developments in genomics, in satellite technologies, drones, citizen science, social media are providing stream of data that open this huge potential for ocean discovery and contributing also to ocean sustainability. So I'll talk about a couple of examples of the kind of insights that this data revolution is providing for ocean health and sustainability. And one of the prime examples come from automatic information system. These are vessel tracking, satellite-based systems that are based on transponders that vessels larger than 50 meters are mandated to carry all over the world. And this is an anti-collision system. It's for safety, but at the same time, it's now providing information on the identities and positions of vessels in a majority of the world ocean every few minutes. And this is the map that you see here. So these data streams combined with machine learning also now allow discriminating different kinds of activities. For example, deep sea mining and fishing, and even different kinds of fishing, as you see here. So the result is a picture, a snapshot and an ability to track to time changes in what happens in ocean ecosystems, far from shore, in places where direct observation can possibly provide this kind of information. For example, this is an image of the fishing effort globally in 1 year, 2016, that was provided through AIS data. So this is really important because for the first time, just in the last few years, now this was unimaginable before, we can do surveillance of large offshore protected areas. This data can inform fisheries management. For example, parallel advances in animal tracking. These are satellite tags that Barbara Bloch's lab at Hopkins Marine Station has developed, integrated with AIS data, have enabled the identification of hotspots of overlap of fishing activity with the essential habitat of endangered and threatened shark species. So here you see the overlap of different kinds of fishing gears that catch tuna, and then in plot B, they catch 7 different species of sharks, including threatened ones. And this overlap now can guide management interventions that can reduce the bycatch of sharks in a targeted way, for example, through temporary closures or gear modification. There's also vast room for advances, for new frontiers. And as an example, this is a project that's supported through the Stanford Data Science Initiative that was developed because of the Data Science Initiative, that is aimed at expanding the capacity of the current vessel tracking system by utilizing planet satellite imagery, high-resolution coastal imagery, to enable tracking of vessels that are too small to carry the transponder or that are dark, meaning that they illegally turn the transponders off. So this is a collaboration that spans ocean science, computer vision, statistics, data science. Very exciting potential. And then also the other frontier is taking this information all the way to promoting change in the water, improvement in the water. And one of the grand challenges that we're still facing in ocean is illegal and unregulated fishing. This illegal activities rob nations of an estimated 26 to 50 billion US dollars every year. They are pretty much the obstacle to sustainable management. Without addressing, curbing this plague, there will be no sustainable management. They affect lives. They are connected to labor abuse, human rights violation. So it's a big problem. So in a paper that just came out today in Nature Communication, the team from Center for Ocean Solutions with other collaborators essentially combined AIS data of fishing effort that you see here on the left. This is 8 million fishing trips, with information gleaned from expert opinion survey about the risk of illegal activity and labor abuse in ports around the world. Basically where these fishing vessels are going to after their trips, as a way of then tracking back the hotspot of illegal activity at sea, where fishing it has a high risk of being associated with illegal activity, which you see there in the map on the right, identifying 3 levels of risk, high, medium, and low. And you see hotspots of risk, for example, of West Africa in the Western Indian Ocean off Peru. So then the team is working with what have been called the key actors, the largest seafood companies in the world, the 10 major seafood companies, because their commitment to curbing illegal fishing can leverage this data to identify risk and work to eliminate these activities or at least decrease them through their supply chains. And then, I don't know how much time I have, but I want to transition to another couple of examples that focus on another problem, which is the open question of what's the contribution of blue foods, the fish, shellfish, seaweeds that are produced or harvested in aquatic systems to addressing food insecurity and malnutrition. So answering this question is very difficult. Data were scarce. And as part of an initiative that is called the Blue Food Assessment, that was co-led by the Center for Food Security and Environment and Center for Ocean Solutions here at Stanford, as well as Stockholm Resilience Center, The team had compiled information on nutritional profiles of blue foods from over 3,700 different species of fish, shellfish, and seaweeds. And so this resulting database of the nutrient content now has allowed analysis and comparison of the nutritional value of different aquatic species, for example, in comparison with terrestrial species. So in blue, you see the blue foods, the aquatic species, and then in green, terrestrial counterparts. And so this analysis for the first time are showing that there are several species of aquatic animals and seaweeds that are major contributors, have really high value in terms of nutrition, particularly in terms of calcium, omega-3 fatty acids, and calcium. And then, so the second question is, where are blue foods consumed? Where there's greatest reliance on blue food, and conversely, what areas might be targeted through policy interventions to action to bring the superfoods that have been identified to address malnutrition hotspots. And so these analyses, again, were enabled by big data that weren't available, accessible, at least in this case, up to a few years ago, and consist in the results of yearly surveys of fish and shellfish consumption in households every year across 50 different countries. So here, these data are often reported and visualized at the national scale, and you can see patterns of and hotspots of seafood consumption. But now access to this data at the household scale enables a much richer picture and shows, for example, that in some countries, high national level consumption corresponds to a relatively homogeneous consumption across the country. But there are instances, for example, India and Peru, where that low national level consumption actually masks very high heterogeneity with hotspots of high consumption and reliance of blue foods in these countries. And so now governments and development organizations can target specific areas with this more nuanced information, for example, with programs like school feeding programs or trade networks that can bring blue foods when, where they're needed. So I want to conclude by highlighting three key components of the challenge of bringing this data revolution, this new capacity to ocean health and to ocean challenges. And this includes continuing to support the collection and access of data in real time and at scale. The second pillar is supporting the analytical advances that transform this data in information, in understanding. And then the third and really important component is to connect the research and education to the need and use through partnerships, for example, between academia, government, industry, and civil society. And so I want to conclude with the optimism that Stanford Data Science and the new school focused on climate and sustainability bring as the perfect environment for what is really the essential ingredient of all of this, which is collaboration across disciplines, across groups, with external partners. And this is really echoing what other colleagues have said before me today. This is the key ingredient to make these major advances that we want and need to make for discovering sustainability. Thank you very much.
Great. Thank you, Fio, and thanks to all the speakers for being so punctual, even a little ahead of time. So our next speaker is Guido Imbens, and Guido is, among other things, a professor in the Graduate School of Business here, but he's also a director of one of the centers. We haven't talked much about the centers within Stanford Data Science, but at lunchtime there will be tables set up where you can be introduced to them. Guido is the director of the Causal Science Center, the Stanford Causal Science Center, SC², which began this year, and I'm told it is definitely the most exciting thing that's happened to him this year. So with that, I will pass it on to Guido.
Thanks. Thanks very much. So where's the clicker? Oh, it's right here. Thanks very much. It's great to be here. It was very nice to see the different talks so far and see all the exciting work people are doing. It's also a little humbling to see that the standards for slides are much higher in some of the other disciplines than compared to economics. So I'm sorry about that. So what I want to talk about is three things. So I just want to briefly talk about what I mean by causality, kind of what in economics and social sciences people typically focus on. And then I want to give two examples of research I've been involved in recently, looking at causal effects in settings where there are some challenges with that. So the first thing about what do we mean by causality? In the end, what I'm interested in is looking at what the effect is of some intervention, of some manipulation, where something is changed in the environment for particular units. And these units can be individuals, they can be geographic areas, they can be firms. But some units are exposed to some intervention, and they could not have been exposed to that intervention. And what we're interested in is the difference between what is in that literature sometimes called potential outcomes. So a very simple example is I may have a headache, I may think about taking an aspirin, and what I'm interested in is what the state of my headache would have been had I taken the aspirin relative to what the state of my headache would have been had I not taken the aspirin. Because the challenge is that I can't do both at the same time. And I can't ever directly measure these causal effects. And so we're going to need to have multiple units, multiple individuals, some taking the aspirin, some not taking the aspirin. And so that then creates challenges because the people who took the aspirin may be different from the people who didn't take the aspirin. They may have taken it by choice, and their headaches may have been worse. And so we need to worry about why some individuals, some units got exposed to the treatment and others didn't. And if we have a randomized experiment, we know exactly how that happened, and we're going to get a lot of leverage out of that because there we know it was purely by chance and it didn't have anything to do with systematic differences between these units. But in social sciences, traditionally, in many settings, we can't do experiments, and we face much more severe challenges in trying to come up with credible ways of estimating causal effects. So some of the things I want to talk about are about the challenges in those settings where we can't do experiments either because they're too expensive, because they wouldn't be ethical, but for whatever reason, we have to make do with observational data. And then the second thing I want to talk about is some of the challenges in experiments in settings that go beyond the simple type of experiments that Fisher and Neyman worked on in the 1920s and 30s, where you can simply divide a population of units into two groups, a treatment sample and a control sample, and you can compare average outcomes. In many settings where people are doing experiments currently, there's a lot of interactions, there's a lot of spillovers between different units, and that creates lots of challenges. So the first example I want to look at is a setting where we do actually have experimental data, but they don't answer the question we're interested in, or at least they don't answer directly the question we're interested in. And so we want to combine that with observational study data, and want to think about systematically combining the experimental and observational data so that we can get answers to the questions we're interested in, where the experimental data are not going to be sufficient for doing that. And so the hope is to both make the experiments more valuable by allowing them to weigh in on questions that they don't directly answer, as well as making the analysis from observational data more credible. And so the specific example here is one from a paper with Susan Athey and Raj Chetty, where we're interested in the effect of a childhood intervention, of having small classes for children on tests on educational outcomes. And so here specifically, we're looking at the effect on test scores in 8th grade. So these small classes were early on in the children's educational careers, and want to see if there's some long-term effects of those. It would have been nice to do an experiment that measured these long-term outcomes, but that would have taken a long time because these are class sizes early on for these children, and we're interested in the long-term effects, but we don't want to have an experiment that takes 10 years to do. So we have two data sets. One experimental data set where children are randomly assigned to small or regular class sizes for which we measure outcomes in 3rd grade. So we have these 3rd grade test scores, and we get credible, precise estimates of the effect of small classes on 3rd grade test scores. Then in addition, we have observational data from a different location from New York, where we also know whether the children were in small or big classes, but now we have outcomes both for 3rd and 8th grade tests. And so what we see is that the results in New York are very different for the 3rd grade, where in the Project STAR experiment, we see an effect of the small classes that improves test scores by 0.2, and that's very precisely estimated. In New York, it looks like the small class sizes actually made test scores worse. And we think that that's probably not true, the true causal effect. We think that's because the children in the small classes were different from the children in the big classes. And so as a result, we don't really trust the results for the 8th grade either. There again, we see this negative effect, but based on these data, I would not think that the effect of small classes on test scores is actually negative. And so what we're looking for is a systematic way of combining these data from this experiment to figure out what the effect is on 8th grade test scores. And so what we do is essentially build a model. So here's a graphical version of that, kind of in the line of, in the spirit of the work by Pearl, where we think the reason we can't simply compare 8th grade test scores in the observational data is that there's some unobserved differences between the kids in the small and regular class sizes that affect 8th grade, that are also correlated with 8th grade test scores. Now, what the key assumption we're going to make, and this is not completely clear just from the graph, but the key assumption we're going to make really is that whatever that unobserved confounder is, whatever these unobserved characteristics are that make that invalidate the comparison in 8th grade test scores for in the observational study, that unobserved confounder is the same for the 3rd grade test scores. And so we're going to leverage the fact that we can actually extract the true effect for the 3rd grade test scores from the experimental data to figure out how to adjust the 8th grade test score results for the observational data, where we don't see them in the experimental data. And so at some level, the starting point is that we think the minus 0.118 is not a credible estimate for the 8th grade, the effect on 8th grade test scores, because we see that for 3rd grade test scores, the observational data don't give us the right answer. We think that the observational data have low internal validity, and we want to use the experimental data to improve that. So to kind of summarize the results there, what we do, the data we have are these 3rd and 8th grade test scores for the observational data and the 3rd grade test scores for the experimental data. The simple raw differences are not very credible, but we're going to adjust them using the experimental data. And what that's going to lead to is estimates for the 3rd grade test score that are now the same as for the experimental data. So you see that the adjusted estimates are the 0.201. And now applying the same adjustment method, and I'll give a little bit more, though not a lot of more detail on kind of how I get to this adjustment, but we adjust the 8th grade test scores in the same way, and that gives us an estimate that is now positive of 0.057. Now, do we know that that actually works? Well, we do. And so partly it's because I actually lied earlier. We do have data for the 8th grade test scores in the experiment. We do know what the effect is there based on the experimental data, namely 0.085. We had not used that data. We put it aside in this analysis and adjusted the data purely based on extracting the unobserved confounders using the 3rd grade data, and then use it to analyze the 8th grade data, adjusting for that estimated unobserved confounder, that gives us an estimate of 0.057 that is actually very close to the 0.085. And so kind of the intuitive version of what we're doing there is we're taking the 3rd grade test scores, we're extracting from that what is driving the bias between the experimental and the observational data. We interpret that as the reason for the difference between New York and Project STAR, just the fact that there's this unobserved confounder that selected the students into the 8th grade and into the small and regular class sizes. Once we've estimated that, we use that as something to adjust for in the 8th grade data, and that gives us this adjusted estimate that is actually very close to the truth. And so let me, because I'm going to run out of time, let me skip the details there, sort of how we actually do that, but it's essentially what I said. We're extracting from the 3rd grade data what is driving this bias, and then we're using that to adjust the 8th grade scores. Second thing I want to talk about is sort of a project, and there's an area where there's a lot of interesting work going on about experimental design in settings where we're worried about interference between units. So traditionally, experimental design was in settings where the units are all separate. Whether I take aspirin or not doesn't affect somebody else's outcome. And so I can analyze units separately. I can divide things into a treatment and a control group. But in many settings where people are doing experiments now, in particular in online settings, there's a lot of reasons to expect that there's interference. Many of the companies that are doing experiments are marketplaces, are platforms that bring together different sets of units, and they structure the interaction between these units, but fundamentally they're exploiting the fact that they're trying to leverage the fact that these units interact. And so, for example, if the company like Airbnb offers some discounts on some rental properties, that's going to affect outcomes for other rental properties, because people are going to switch between some renting one property versus another. Or if a rideshare company makes it more attractive for drivers to work at a particular time, that's going to affect drivers who are not in the treatment group, because they're going to face more competition during those times. So in all those cases, we need to address the fact that there are spillovers, and these spillovers are often of first order importance. Now, so here's one way, and I actually put some references there from other people here at Stanford. We also have developed new experimental designs to deal with some of these issues. But so here's one particular way of dealing with that in a setting where instead of having a single population, we actually have multiple populations. So suppose we're looking at a setting where we have customers and products. And we could do an experiment, and we're interested in the effect of giving customers more information about the products when they search for these products online. And so one thing we could do is simply do a product experiment where we randomize products to the treatment group or the control group. The complication is that people may switch between treated and control products, and so simply comparing treated and control products wouldn't tell us what the average effect would be if we expose all the products to the treatment versus none. We could also do a customer experiment where we randomize the customers to the treatment group or the control group. Again, there could be spillovers there. And so one way of trying to deal with that, to address that, is to think of the assignment not as a factor, not as something where you randomize the population of customers or you randomize the population of products, but as a matrix where you think of the assignment as something you can apply to the product-customer pair. And of course, that's not possible in many in sort of brick-and-mortar stores where you can't give two people the same experience when they visit the store at the same time. But in online settings, we can give different information to a particular individual for a particular product relative to the information we give to a different customer for the same product. And so we can choose the treatment at the level of the customer-product pair. And so from that perspective, you can think of customer and product experiments as special cases where a customer experiment also has this assignment matrix, but every column in that matrix is the same. And a product experiment is an experiment where every row in that matrix is the same. But kind of once you set it up that way, it's sort of clear you can go beyond that, and you can actually decide to systematically create sets of product and customer pairs that you expose to the treatment. And that's going to give you the ability to compare different sets of control groups and get a handle both on the direct effects of the exposure to the treatment, as well as the spillover effects, as well as the effects coming from the interference between units. And so here's a very simple version where you can compare this green control group with the red control group and learn about spillovers within customers. But you can do much more complicated things. And so here, for example, a setting where we first split the products into two groups, and we do a customer experiment for one set of products and a product experiment for a second set of products. And that's going to give us a lot of leverage to figure out what the spillovers look like and how we would actually predict what would happen if every pair was exposed to the treatment versus no pair. So let me stop here and not get into trouble for going over time here. But thanks very much for inviting me to talk here.
Great. Thank you, Guido. So we now have about 12 minutes or so until the lunch break. And so I'm going to ask the four speakers to please come up and sit in front of a mic here. We have 3 mics. I'll put one back in a second. And invite questions. Is Yuri still here? No. Okay, then we only need 3 mics, I guess. So I guess I have a question, as you guys think of questions. I don't see any hands up, which I guess is for all of you in some ways. And going back to what Sandrine put up, kind of jokingly about why do something simple when you can do something complicated. But there is, I think, a general trend, which is that as these fields get more and more sophisticated, in terms of the methods fields like statistics, machine learning, and there's a lot of students in the crowd that are those types of students. They're needing to innovate on the methods, right? They're needing to find something unique about the data that will be, interesting for their communities. At the same time, as that frontier gets further and further, it becomes harder and harder for this, students like Fio students or my students to be able to implement, what was new 2 or 3 years ago in that field. So I'm just wondering, in all of your experiences, how have you advised students to find that sort of happy medium between doing something that's useful to the application area and doing something that is innovative? And I guess, it would be a different question from your different perspectives because you're all in different fields.
So, I'm happy to make some comments there. So in economics, there was a time sort of in the 60s to the 80s where suddenly there was concern that people started using more and more complex statistical models for analyzing data, to the point that people became very concerned that the results from those data analysis were not very credible. There's a very famous paper in economics with the title, "Let's Take the Con Out of Econometrics," that was all about the fact that people presented these results of these analyses kind of with great confidence and with very small standard errors, where really they were not very believable. The specific example was one where people looked at the deterrence effects of the death penalty, and people came up with these estimates that every execution would save 17 lives, plus or minus 1. And so it was clear that those results were very unreliable. And so in economics, part of the trend since then has been to rely on simpler methods and more credible methods, and be a little bit more modest and try to actually get better data and get more credibility that way, and away from some of the very sophisticated, complicated modeling.
Well, I think that's a really interesting question, an important question, David. When it goes back to the students, I think it really depends on what their interests are. I think there's room for all of these endeavors, but if you are a student who is genuinely interested in working in applied statistics or data science or whatever you call it, I think you should start from the question and not think I have this method, I want to fit it to a particular problem. It's the coolest theoretical method, all these bells and whistles. So it really depends on what the context is, what they're trying to do. So there's room for students who want to do theoretical research and try to work on sophisticated methods and prove optimality properties and whatever on it. But I think you need to really think about what question you're trying to address, what your goals are.
And just to follow up on that, so your communities, I mean, I think we'd already said this for econ, but your communities have become more receptive over time to students who are focused more on applied work.
Oh, that's a really good question. I was on a panel at the ASA, sorry to jump in. The ASA, the Joint Statistical Meetings last year, just about that, about applied statistics in academia. I think it's taking time. Gradually, we're more open to having junior faculty who are working on applied problem. It's harder, I think, to assess students and young faculty working in applied statistics, just because of the breadth of the discipline. We don't have the framework necessary for thinking about that. I think the same goes also for research in statistical computing. I think it's still somewhat misunderstood how to handle that. I think we're more open about it, but we have a long way to go about it. I've seen this when we hire, in faculty hires, it's very easy to find a star probabilist. Everybody agrees on that, and it's like, yes, let's hire that person. When we try to hire an applied person, it's much harder to come to a consensus about the standards we want, the expectations. So some room to go, some improvement.
Well, my field is marine ecology. Marine ecology, my training went through a phase of huge enthusiasm for experimental manipulations. Experimental design courses, everyone had to do experiments, and experiments became more and more complex and more elaborate and more rigorous, but also addressing questions like, what is the movement of that limpet over now? Because by necessity, then you focus on systems that are amenable to that kind of work. Those are not necessarily the most important questions. So I think that I agree that question is key. Often the interesting questions come from cross-pollination from another field. For example, again, in my field, in ecology, meta-analytical techniques and then advances in meta-analysis that came primarily from medical and social fields have really revolutionized the kind of questions that are asked in ecology, synthesis of experiments, then these in turn have really informed what experiments need to be done, what design. So that, I think that aspect. And then the last thing is that I think that there is a great need for specialists as well, those that really dive in deep and decide that that's their field, and then really make discoveries that have value in itself, or as part of teams bring that. So in part is deciding what is most interesting, I think, to students. But those are the things in my experience that I've learned through time.
Okay, great. We got a question here.
So I want to follow up on Sandrine's comments about exploratory data analysis and the importance of it. One of the things that we've seen in psychology, through this kind of decade of reproducibility crisis, is that students have become incredibly afraid to explore their data at all. Exploratory data analysis has become like a bad word, to the degree that it's very easy to get garbage in and garbage out. But at the same time, you do have to worry about exploration and the degree to which it can become circular in your inferences. So I wonder how you think about kind of threading that needle.
Between exploratory analysis or just
Between exploratory analysis and p-hacking or other questionable research
P-hack.
practices.
p-hacking, you said.
Yeah, p-hacking or
p-value right.
circular data analysis. Like how do you ensure that you do correct exploratory analysis, but make sure that it doesn't sort of negatively influence the quality of the inferences in the end?
Oh, I see. You mean just data snooping and...
Exactly.
Oh, right. Yeah. I think you have to keep in mind the goal of the study. I mean, in our case, the exploratory data analysis just is basically to understand what's going on with our data. And oftentimes we find problems with the data. In the genomic context, that's what it is. So it's not so much to dictate what conclusions we're going to make for the data, but it's finding mislabeled samples. That happens all the time, or finding technical biases and so on. So it's not really to dictate what hypotheses we're going to test or what conclusion we're trying to. It's just more, what's going on. Understand what are the features of your data.
It's a really important point
So there's a distinction. I can see that. Yeah. And just quickly, I was really happy to hear your comment about having experts. Because I think that's a danger we have in data science. And I go back to my experience in computational biology from 20 or so years ago. People would apply the same label of computational biologist to me, where my expertise is in statistics, and my colleagues whose expertise is in genomics. And we contribute very, very different things to a problem. I think you need teams of experts in different disciplines to solve these problems, not one person that can do it all. So I think depth in whatever discipline you're interested in, depth in the domain, depth in computing and statistics. I think that's the key, be able to work together on these projects and find a common language, work at the internet. So thanks for mentioning
that's true
that.
in all disciplines, and that's where data science can just be very helpful in fostering these collaborations and bringing these specialists into the picture.
We have time for maybe one or two more questions. One more.
So I had a very quick question. First of all, thank you all for the very nice talks. I'm curious to hear your thoughts about the role of open science in all three of your fields. In particular here, I'm thinking about how the research you do is directly tied to behavioral choices or medical or policy choices being done, and how open science can actually be somewhat of a disadvantage. So for example, if you're trying to create models that can track illegal fishing, the more open you are about how those models work, the easier it is to circumnavigate them. Or this recent Nature publication talking about how models used to find drugs that are non-toxic can be reversed to recreate like famous neurotoxins. So how do you balance your obligation as a scientist to create open, reproducible research with some of the moral obligations that can come from the impact that open research can have on the world around you?
A nice simple question to finish us off.
No, that is a great question, and I'm very excited about the panel on ethics that we have today, because I think that those are the questions in illegal fishing or anything that involves human rights and human lives and health and all that is becoming increasingly important in marine science. No? A decade ago, someone like me would have not even thought about this, but now it's what we do daily. And so on one hand, the access to data enables this kind of analysis, but then it brings all sorts of problems and risks. For example, highlighting the risk of illegal fishing on certain vessels can put a huge risks the laborers that are on those vessels. So those ethical issues are at the very center of discussion in this field right now.
I think they would all agree it's a hard balance, and you have to really be thinking about that balance, because they're definitely, you don't want to just say blindly that you want to always be completely open. At least that's my experience. So that is also a good segue to the need for collaboration and the need to think about these questions of causal inference, open science. It's a good segue to the lunch, because we will have the 4 centers all represented, and you can go and find more about them, and that maybe is what Chris is coming to encourage you to do. I don't actually know where lunch is, so I'm going to hand it off to him. But let's thank again the great speakers.